saw it in the...
Oh.
Ooh, sorry, that was me.
Full screen mode. Okay, thank you. It's working?
There. Almost there.
Almost there.
Maybe we should unplug it again. Plug it back in.
Not working.
No worries. Let me just try this. We get out of it, we put it on. It worked all the time, so it will be fine at some point. How did you get this to?
Yeah, just view full screen. I'm not sure because I'm not used to Windows.
Yeah. Okay.
We have a technical problem.
What could he be doing wrong? Do you have a computer?
I have it, yeah, but I don't have actually the HTML.
All right.
I don't know what you can do.
Drup. Huh. That's funny because this is the computer they gave us. This is the one supposed to work. Yeah.
Can we borrow the computer from anyone, or?
Do you have it on your USB stick?
Yeah, I have the USB. Not sure why it's not working anymore. Okay, let's see this. Oh, okay, thank you. Thank you for helping. Oh, yeah, sure. It's working, maybe. Oh. Yeah, okay, we made it. Yeah, full screen, and then I can start right away.
So it's always good to have an expert do this. So the next talk is by Francesca Petralia, talking about a new method for constructing tumor-specific co-expression networks.
Hi, yeah. Thank you so much. Thank you for your patience, because we had this technical difficulty. Okay, so this is a joint work with other people. We have Pei Wang, Arthur Yan, Ji Peng, Li Wang, and Jun Zhu from the Mount Sinai, and then Ji Peng from UC Davis. So I'm going to start with the problem setup. So everybody knows that tumor tissue are a mixture of tumor cell as well as non-tumor cells. So we have a variety of cells that infiltrate the tumor tissue, like stromal, B cell, T cell, macrophages. And it's important to take this into account, the tumor purity, when constructing co-expression network. Why is that? So let's assume that we have 3 genes over there. So we have these light blue genes that are differentially expressed between non-tumor and tumor cell. And then we have that only these 2 genes are expected to be co-regulated, co-expressed in tumor tissue. But then when you don't consider the tumor purity heterogeneity and you construct a co-expression network, you are very likely to detect an edge between these 2 guys only because they are differentially expressed between different tumor cells. So you have this mean shift across tumor and non-tumor cell. Instead, when you take into account the tumor purity heterogeneity, you would detect an edge only between the 2 guys that are expected to be co-expressed in tumor cell. So you want to take into account the tumor purity to reduce the false positive in detecting those associations that are you detect only because there is this differential expression between tumor and non-tumor cell. So tumor purity has been estimated using different data types. So in the last years, many methods have been proposed. Some of them actually infer tumor purity from next sequencing data, others from copy number variation, methylation data. Then there is this nice review paper, that basically compare different methods on TCGA, on all TCGA data sets, and they prove that basically most of the algorithms converge in the estimation of tumor purity. So that's nice to know. But despite this progress, to our knowledge, there is no algorithm that takes into account tumor purity when constructing co-expression network, and this is what we do. So we propose this tumor-specific network, TSNet, that first estimates tumor purity level, but of course this is not actually the main point of the algorithm because you have a bunch of algorithms doing that, but also take into account the tumor purity to estimate co-expression network. So basically you will be able to estimate a co-expression network for the tumor and non-tumor component. So what we do is very simple, and actually people have been using this model quite a lot. So we have AXGN is the gene expression of gene G and sample N is modeled as a mixture of latent variables where pi n is the tumor purity for sample n, and then you have YGN that is the gene expression that you would measure in tumor cell alone, 1 plus 1 minus pi n, and you have this other latent variable ZGN that is the gene expression that you would measure in non-tumor cell. So what we do is that we assume that we have some prior estimate of tumor purity that you can derive based on other algorithms, like you can use absolute that infer tumor purity from copy number variation data, and then we consider this prior information in our model and we derive a final estimate of tumor purity. Then considering tumor purity as fixed, those pi n, we basically learn the inverse of the precision matrix of the latent variables. Other methods have been considering those latent variables, Y1 to YG, to be IID from some normal distribution. Instead, we basically model the covariance structure of those latent variables. And learning the inverse of the precision matrix for the Ys and the Z is going to give you the co-expression network for the tumor and non-tumor component. So this is basically the model. So this is what I described now. And then we have the latent variables that are the gene expression that you would measure in tumor and in non-tumor cell that are modeled with the multivariate normal. And we want to infer the inverse of the covariance matrix. And then we have some observed tumor purity, HN, that is modeled as a beta, with some parameterization such that the expected value of the observed, this prior tumor purity, is going to be equal to the actual tumor purity, pi n, that is unknown. And then we have this penalized likelihood where we use the L1 norm for both covariance matrices for the tumor and non-tumor component. And for each penalty term, we have a tuning parameter that is controlling the sparsity of the network. So by varying the tuning parameter, you're going to have larger networks. So this is basically a lasso penalty. So it's just it's a standard model. So I will go through some synthetic data. Okay, so for the synthetic data, so the first synthetic data experiment is very easy because we sample just from the model. So we expect to do good. I think so, yeah. So we just sample from our model, and then we want to compare our performance with the standard graphical lasso. That basically, the standard graphical lasso is just the graphical lasso applied to mixed gene expression, and is going to infer just one network. And then we basically try to understand how that estimated network overlap with the tumor network and non-tumor network. And then we apply our TSNet. So to sample just briefly, so to sample the tumor purity, we use the distribution of tumor purity in TCGA breast cancer data. So we sample the tumor purity from that density. And then I'm going to explain a little bit later how we draw the networks. But anyway, we draw it from the power law density. And then we sample the latent variables. So we sample the mixed gene expression data from the mixture model. And then we sample the observed purity. We consider 10 replicates data set containing 200 observations each. So this is the results for tumor purity estimation. Actually, I didn't mention that for the observed purity, we use different value of delta. Delta was a parameter is controlling the variance of this beta distribution. So when delta get low, you have that the prior tumor purity diverge from the actual purity. So we actually have the results for different level of delta, different level of noise. So as the level of delta increases, you have that the observed, so the prior tumor purity correlate better with the truth. But no matter how the level of noise is, we can actually, so this plot shows the correlation of the estimated purity with the true tumor purity for different level of delta. So as you can see, for different level of delta, we can recover pretty well the true level of purity, no matter the level of noise. So in this box plot is over 10 replicates data set. So this is actually for tumor purity estimation, but as I said, this is not our main point. Then I show you results for the estimated network. So here for the network generation, we consider 1K node, 1K genes, and we divide the genes into 4 disjoint clusters, each sample from a power law density. So basically, we follow the strategy of Danaher et al. So for the first simulation scenario, we have that the tumor and non-tumor network has some overlap. So 2 clusters are exactly the same. So the standard graphical lasso should perform nicely here. And then for the second simulation scenario, we just randomly generate each subset, sub-network. So we don't force any overlap between tumor and non-tumor network. So for these, actually, the standard graphical lasso might have some problem. So this actually is the average of ROC over 10 replicates for different simulation scenario. We have for dimension 1K, sample size 200, dimension 1K, sample size 400, and we have results for TSNet, normal and tumor component, tumor network, and MixNet. As you can see, for each false positive edges, we actually have a higher true positive. And basically, the true positive rate actually double as we double the number of observation. As we actually draw the network independently, so we don't force any overlap across tumor and non-tumor network, as you can see, the MixNet poorly infer those networks, and is actually pretty intuitive. Okay, so also, so as I said, we have this tuning parameter, and by varying the tuning parameter, we get a network with different size. We can select a network using a BIC criteria. So we just select the network that minimizes the BIC. So we did that, and here is the box plot of AUC over different replicates. The first row is the true positive rate. Second row is the false positive rate. And these are different simulation scenarios, varying the number of nodes and the number of samples. And as you can see, the TSNet is the red, and MixNet, that is the standard graphical lasso, is the blue. So we basically have higher true positive and lower false positive for all simulation scenario. So then what we did is to compare also TSNet versus DMix plus graphical lasso. So DMix is this nice algorithm that can deconvolve the mixed expression data into tumor and non-tumor component. So basically, you get the X, you measure the X, and they deconvolve the signal into Z and Y. So what we did is to compare our model with DMix, applying DMix, so you get the Ys and the Z, and then you apply basically the standard graphical lasso on the Y and estimated Z. Actually, we need to mention that DMix consider Y and Z to be log2 normal. So we basically sampled the data using the log2 normal, so their method. So we are actually estimating the network under model misspecification because our model is just a Gaussian model. But anyway, okay, so we simulate the data from their model. We consider 25% of the genes differentially expressed between a tumor and non-tumor component. We consider 25% of the sample from the non-tumor component, so some of the samples have purity equal to 0. And then we compare our approach to DMix plus graphical lasso. So this is the performance on just one synthetic data scenario for tumor purity estimation and network estimation. So this is basically the correlation between true level of purity and estimated. And as you can see, we get closer to 1. So this is, again, the box plot over 10 different replicate data sets. And then for network estimation, again, so we have the mix and TSNet in red for normal and tumor component. And for the normal component, you can see that we can more accurately estimate that component because we have a lot of sample, 1/4 of the sample are from that component. So that's why we have more information for that component. But still, we do better than the mix plus standard graphical lasso. And then we applied our method to TCGA ovarian cancer data. So this is the purity estimation. So we compared our tumor purity estimates with the one from estimate and absolute. Estimate is this nice algorithm that infers tumor purity from RNA-seq data. And then absolute is this well-known algorithm for the estimation of tumor purity from CNV. And then we compare the tumor purity with the tumor purity from leukocyte signatures that have been actually used to validate those signatures. So as we can see, we correlate a lot with estimate, and actually this is expected because we derive the tumor purity from the same data, RNA-seq data, but actually we slightly better correlate with the tumor purity from leukocyte signature. Okay, but anyway, this is not our main goal, the tumor purity inference. Then we basically selected 1K genes, and we applied TSNet on these genes to learn the co-expression network. And we compared the performance with the standard graphical lasso that basically identify only one network. So basically, this is actually the biggest subcomponent of the network. And here you have in red the TSNet tumor-specific edges versus in blue the edges shared between TSNet tumor and MixNet. So in summary, basically, you can find more information in the paper, but basically we showed that our TSNet, in our TSNet, some tumor-related genes and tumor repressor like HIC1 was a hub gene in our networks, but not in MixNet, using the standard graphical lasso. Actually, HIC1, that is a tumor repressor, was a hub gene in both a TSNet normal and TSNet tumor. But actually, by looking at the neighborhood of this gene in both networks, we figured out that different biological processes were involved. For TSNet tumor, we had pathway, this neighborhood was enriched of DNA damage response and other tumor-related pathways. Instead, for TSNet normal, we have the ribosome, extracellular matrix, collagen pathways that were enriched. So basically, even if this gene was an up gene in both networks, different biological processes were involved. Also, we did an enrichment of the networks, and what we show is first MixNet. So these are basically the number of enriched category using the molecular signature database from the broad. So we can show that MixNet, so the standard graphical lasso, overlap a lot with TSNet normal. First of all, MixNet is the one with more edges, and this is expected because, as I said at the beginning, if you don't take into account tumor purity, you're likely to detect an edge between genes that are just differentially expressed between tumor and normal. And also you will detect, of course, the edges between genes that are involved in the same biological process in tumor cell. So, but you're very likely to detect more edges because you get a lot of false positive. And as you can see, there is a lot of overlapping with TSNet normal. And if you look at the pathways enriched for TSNet tumor, there are a lot of cancer-related pathways. Instead, MixNet actually has some cancer-related pathway, but also stromal-related pathway, ribosome-related pathway, ECM, so that are already captured by the other TSNet normal, basically. So yeah, there are more details in the paper. So this actually in summary, so what we did, so we proposed this algorithm that can estimate a network for the tumor component and non-tumor component, and this is the first time that this has been done. Okay, so actually this algorithm was driven by TCGA data, but it's going to be applied on CPTech data soon because I'm involved in the consortium. And so the CPTech data is a proteogenomic data, so we are going to get the gene expression as well as protein expression for different cancer types. And one of the cancer is kidney, and it's well known that kidney is the least pure tumor across all the solid tumors, so we are pretty excited to apply this method there and also borrow information across proteomics and gene expression data in order to do so. So there is also some model development that is expected. And also for kidney, there is this severe immune infiltration, and we will try also not only to characterize tumor versus non-tumor, but different cell types. Of course, you have an identifiability issue there, but there are some ways to handle that. Thank you so much for the attention.
Questions? I think I have one. For some cancer types, there are normal control samples, so they're not tumor samples, but they were, during surgery, they were taken out from the normal tissue. Mm-hmm. If you derived a network on those, would it look the same as the one you derived from your mix?
It's not exactly, it's a great question. Actually, it's not exactly the same because the cell that infiltrate the tumor are a little bit different than the normal cells. But usually for the identifiability problem, we consider the normal as the same as the cells that infiltrate the tumor, even if this is not really correct. It's a big assumption, yeah.
Hi, very nice work. I was wondering how you think your method would currently work or what you could do to extend it to work in the situation where you have metastatic tumors, where depending on the site of the metastasis, you could have very different types of cells, normal cells contaminating the tumor cell.
Oh, that's a very good point, actually. That would be nice to extend this method to metastatic cancer as well. We didn't think about that yet, yes.
Let's thank the speaker again.